Hello and welcome to Nikolai's genetics lessons, and today's problem is Arabidopsis thaliana is a small well-studied plant, responds to cool temperatures by synthesizing FAD8 proteins. FAD8 is a fatty acid desaturase enzyme which converts single bonds between two carbons in free fatty acids into double bonds. Why is this enzyme made when the plants are in cold environments? And I will start my explanation a little bit from far, using some of the gases as example. For example, carbon with 4 hydrogen around it. We know that this is methane. If we connect two carbons and also put hydrogens around them, so each carbon atom needs 4 bonds. So 1, 2, 3, 4, and this carbon also has 1, 2, 3, 4 bonds. So this is ethane. This is also flammable gas. And let's continue. For example, we have 3 carbon atoms. Once again, each one would be surrounded by hydrogen atoms, and we call this is also going to be a gas. We call this propane. And if we add one more carbon, so 4 carbons, I will explain you later why I'm using this as example. So also we have to add hydrogens, and we call this gas butane. We use this gas, for example, for barbecue. And starting with 5 carbons, 1, 2, 3, 4, 5 carbons. Now this is going to be a liquid. Once again, we have to add hydrogens here, and some of the carbons would have bonds to 3 hydrogens and some to 2 hydrogens. And these carbons at the ends would have 3 bonds to hydrogens. So once again, this is pentane, this is liquid, and this is also flammable. And every time we would add 1 carbon atom, we are going to get a flammable liquid. And for example, octane would be something we use in our cars to drive our cars. So as you see, such molecule provides a lot of energy, so it stores energy in its bonds. So we use as humans also energy stored in chemical bonds. And for example, if we delete this hydrogen here and instead add 1 oxygen here and alcohol group here, we are going to get free fatty acid. And this is what we use for storing energy. And plants also have this free fatty acids. When we see that all the carbons would have single bond, we call such a fatty acid saturated fatty acid. But if we would see a double bond between 2 carbons, we call this unsaturated. So what basically FAD8 enzyme makes, it desaturase enzyme. So when it removes 1 hydrogen atom, at the same time it creates a double bond between 2 adjacent carbon atoms. If you take a look at this picture, you would see almost the same molecule but now, as you see, these molecules arrange into one molecule that we call triglyceride. So it consists of three parts. One is glycerin, another part is carboxyl group, and fatty acid. As you see, some of them may have a double bond, and what significance it makes, for example, if all carbons would have a single bond, as you see, such molecule would be more densely packed, and usually we can find such molecules in fats, specifically in animal fats. And when we have double bond, as you see, now this molecules bends, and it's not anymore so densely packed, and we usually find such monounsaturated molecules. It also can be polyunsaturated fatty acid if double bonds also would be between other carbons. So what this creates, it creates more space between molecules, and such molecules would be liquid at the room temperature, and we call such molecules oils. Usually it is vegetable oils, and molecules that is solid at the room temperature we call fats. So usually animal fats. But if you would think that animals has only saturated fatty acid, this is not quite so. For example, let's say that this is going to be saturated molecules, fatty acids, and this is going to be monosaturated, and this is going to be polysaturated molecules that we can find in animal fat. And if we compare with, for example, oils, vegetable oils, we also can find that saturated fatty acids. We can find monounsaturated fatty acids and polyunsaturated fatty acids. So as you see, both animals and plants have all three types of fatty acids: saturated, monounsaturated, and polyunsaturated. Just the balance between these three would makes whether we consider it as a fat that is going to be solid at room temperature, or if it is going to be liquid at the room temperature, then we consider it as oils. But once again, this balance between plants and animals is not stable. Even between, say, plants, we can find different variants. For example, in other plants, we may find just a little bit of saturated fatty acids. We can find not so much monounsaturated, and we can find that majority would be polyunsaturated fatty acids. And more than that, we also may find that in some plants this balance can change due to, for example, weather factors. In autumn or early in the spring, where it is cold, as you see, this enzyme FAD8 would add double bonds between carbons and may convert saturated fatty acid into unsaturated fatty acids. So under the low temperature, such fatty acid would perform better and won't solidify. So once again, a FAD8 fatty acid desaturase enzyme would work on the free floating fatty acid, so this part of the triglyceride, and would add extra double bonds. What the part of the plant produce energy, and this is a chloroplast. So we can find this free floating fatty acids, and this desaturase enzyme in the chloroplasts. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.